Is your mic on now? Should we start? Oh, so maybe I'll wait a few seconds before. Are we good? Okay. Then welcome everyone. It's my pleasure to introduce Dr. Yatish Turakhia, who is an assistant professor in the Department of Electrical and Computer Engineering at UCSD, with affiliations also in the Department of Computer Science and Engineering and the Bioinformatics and Systems Biology graduate program. So prior to joining UCSD, he was a postdoctoral scholar at the Genomics Institute at UCSD. Dr. Turakhia earned his PhD in electrical engineering from Stanford University in 2019 and his bachelor's and master's degrees in electrical engineering from the Indian Institute of Technology in Bombay in 2014. He's a recipient of the MIT Technology Review's Innovators Under 35 Award, Hellman Fellowship, Jacobs Early Career Award, Amazon Research Award, a Media Graduate Fellowship, and multiple paper awards. And of course, many of us know him from many of his influential works, such as Darwin, and with other works that he did with, of course, Bill Daly and with other collaborators. So it's really my pleasure to have you today, Dr. Yatish, and with this, please take it away.
All right, thanks, John, for the kind introduction and also for inviting me for this talk. It's a great pleasure to be here. I guess there's a lot of overlap in the works that the two groups do. So I'm really looking forward to interaction both during the talk and afterwards. So with that said, maybe as John introduced, so I have been an assistant professor at UCSD for the last 4 years. And I started an interdisciplinary lab there with a research mission to develop parallel algorithms, software, and domain-specific hardware accelerators, which enable faster and cheaper progress in biology and medicine. So that's been our focus for the last 4 years, and maybe even beyond. And to realize that mission, we are actually deploying a number of solutions, parallel computing solutions, which span from high-performance computing to accelerating applications on GPU processors, to also developing accelerators for FPGAs and also custom chips. And I'll be presenting some of the tools that we've developed in the last 4 or 5 years in each of these domains, as you will see that it spans all these different solutions. I'll talk about most of these tools later during the talk. But the reason why we are so excited about this particular research area is because we really believe that genomics is at a very interesting inflection point, particularly for us computer scientists. And there are 2 reasons for this. One is that it has become really cheap to generate massive amounts of genome sequence data with the help of DNA sequencing technologies, as many of you would be already familiar. So the idea here is that genome sequences are really the blueprint of life. And the more data we generate, the more we understand how these sequences encode for various functions and phenotypes, right? So it's become really cheap to generate massive amounts of data, but in parallel, we have also developed technologies which allow us to relatively cheaply edit genome sequences and study the effects of those changes. And examples of these include CRISPR-like gene editing tools. And combined, this has a very powerful implication. What this means is that we are able to generate massive amounts of data, but at the same time, we can form hypotheses using computational techniques developed by many technologies that we use in computer science, including AI and others, to form hypotheses about what changes in our genome sequences would result in what function. And now it's become easier to, for example, go and experimentally validate it with the help of some of these gene editing tools as well. So today, as my title suggested, that I'm going to be talking about some of the new waves of genomic data, which we foresee as going to be posing important computational barriers in the future, and which will, and if those computational barriers are lowered, we would go a long way in discovering new biological and medical insights. And some of these waves include an emerging field of pangenomics that I'll talk about in much more detail later in this talk. But the idea here is that we will be in future sequencing lots of individuals from single species. So it's a population of genomes that we sequence from just a single species to study how different genetic variants code for certain phenotypes, right? Then there's another wave of genomic data that we are seeing, which is that in pathogen genomics. So here the idea is that we sequence the different pathogens that may infect humans to study how, for example, they might be mutating and what sort of mutations lead to increased transmissibility of that pathogen, for example, or lethality in some cases as well. Similarly, we are seeing, so you will see this pattern that different subfields of genomic data, any subfield that you pick, you will see that they have this, sort of an exponential growth pattern, which I'll also show in terms of plots. But another such field is that of comparative genomics. So we are not just sequencing individuals from a single species, but we are also trying to sequence many species at once, so as to understand how different species have evolved and what makes certain species, for example, humans, what in their genome makes humans and different from other related species such as chimpanzees and gorillas. Similarly, we are seeing many applications in metagenomics on the rise. So we have been doing a lot of work in sequencing the different microbes, for example, that are present in the gut, which helps in developing, for example, probiotics, or sequencing just the organisms or the microbes which are present in the soil, which influence both the health of the soil and its fertility, or in some cases sequence sewage or wastewater data in order to understand, for example, what sort of mutations might lead to antimicrobial or monitor mutations that lead to antimicrobial resistance in bacteria, or study which different variants might be under circulation in any given geographical area. And look at any of these subfields, and you will see that the data in this subfield is pretty much on an exponential rise in the last decade or two. And as a result, the bioinformatic tools that we might have used in some of these fields, maybe let's say 10 or 20 years back, in most of these cases, they seem to have difficulties in keeping up with the exponential rate at which the data seems to be growing, right? So this is sort of the problem that we are really interested in. How do we lower such computational barriers and enable new applications in this field, right? Enabled by the scale of the data. So in this talk, I will be covering all these different waves of data one after the other, and presenting some of the work or the tools that we've developed, bioinformatic tools that we've developed in these specific subfields. But in addition to that, I will also present some of what you will notice is in all of these applications that there are some fundamental techniques that keep recurring. And those techniques deal with the analysis of genome sequence data, obviously. So two of these fundamental methods that you will see recur over and over again. One of them is what is called the alignment problem. So here the idea is that you're given a set of evolutionarily related sequences, for example, the 8 sequences which I've shown here. And with the problem of sequence alignment, essentially what you're trying to do is to identify which characters in these sequences correspond to each other, right? So generally, what you're trying to optimize for is the sequence similarity. As you align them, you are basically introducing gaps in different sequences and ensuring that in different columns you see that the matching characters typically line up. And then you have another fundamental method that is very popularly used or very widely used in bioinformatic applications and in biological sequence analysis. That's that of phylogenetics. And the key goal here is that you are trying to understand the relationship between different sequences, how these sequences are evolutionarily related. So the sequences which are more similar will tend to group together in this tree-like structure. And the reason for that is that this is sort of a family tree in which the sequences which are derived from individuals that are more closely related tend to be grouped together than the ones which are more distant, right? So it's sort of depicting a family tree-like relationships between different sequences. And so I will also present some of the tools that we've developed for acceleration of these fundamental methods. So I'm going to be talking about applications of these alignment and phylogenetic approaches. I'll cover those, but also we touch upon like how do we fundamentally develop acceleration techniques to tackle these specific kernels as well. So I'll cover both of these topics. But let me, before I move on to the main applications, let me quickly acknowledge the many collaborators that I have, both at UCSD, UC Santa Cruz, and some members in the industry as well. This includes, for example, Russ, Angie, Alex, Pratik. All of these are from UC Santa Cruz, who I closely collaborate with and have worked on since the start of the pandemic on various applications related to pathogen genomic surveillance. But also many of the funding sources that we have. This includes the CDC, Hellman Fellowship, and some of our industry partners, EMD and Amazon included, right? And so there are many people involved in the work that we do, and the students who are leading the projects, I will acknowledge them separately when I talk about the projects that I discuss. So I'm going to start with the applications first of these bioinformatic tools, like the end applications that we worked on, just to inspire what all you can do with some of these bioinformatic methods that we've developed. And I'll start with one particular application, that's that of pathogen genomics, and specifically talk about the USHER toolkit that we developed for this application. So I think most of us are familiar with what went on during the pandemic and sort of have this evolutionary picture in mind about how this virus, as it spreads to more individuals and infects them, it's actually also acquiring mutations, right? So that's the picture that I'm trying to depict here. So they are colored based on the variants that get created. So it started with this particular variant A, and as it spread, it may mutate or not. So in this case, it hasn't. But as C divides into F, it may acquire new mutations as a result. So this is how variants get formed. That as the virus is infecting, you acquire mutations at random, and some of these mutations will actually lead to increased transmissibility of the virus and improve fitness in general, right? So this is a picture that we don't tend to see. Like, we don't exactly know how the virus is mutating, and we don't get to see that in real time, for example, right? There's no technology that can enable that, but we have a good proxy for it, and that's that of genome sequencing, right? So as we get more and more people infected in the population, what you could do is you can sequence the pathogen that has infected that individual and collect those sequences. So this is what I'm depicting here. So you can think of these 6 sequences as being sequences derived from 6 different infected people in the population. And of course, what you will notice is in these sequences, there might be some differences, typically arisen by the mutations that might have occurred as a result of this replication process. And the goal of the surveillance efforts, typically one of the key techniques that we use is to infer the phylogenetic tree relationship among these sequences. So the idea here is that the sequences that we've collected, they are represented at the tips of the phylogeny of this particular tree like structure, and the ones which are more closely related tend to be placed together. So if you look at it, this phylogenetic tree is sort of a representation of the transmission pattern that you see, not exactly, but it sort of informs us like how the transmission might have taken place between different infected individuals. And so this phylogenetic tree-like structure typically becomes the basis of studying the different evolutionary and transmission dynamics of this pathogen. So it's actually a core method that we used in all of the genomic surveillance efforts, right? So one particular application of inferring this phylogenetic tree is that of naming different variants. So you might have heard during the COVID-19 pandemic that there were variants circulating like B117 or XBB and so on, right? So these are actually scientific names that people use for referring to different subtypes or variants of the virus that evolved. And essentially what this nomenclature does is it actually is derived from the phylogeny that we've inferred from it. So it's sort of like the address in that phylogenetic tree-like structure. So what that means is if you have the sequences collected up to a certain date, let's say, and they are all placed in this phylogenetic tree, then originally what happened is, let's say 2 lineages evolved, A and B, and then within B, you had many subvariants that evolved. So for example, B led to B1. So that becomes like, a sublineage of lineage B, and similarly B11 as a sublineage of B1, and similarly B117, that of B11, and so on, right? So this type of nomenclature was widely used. It was in fact first proposed within the first few months of the pandemic as a bioarchive preprint, and it's referred to as like the Pango nomenclature system. And since then, it has become widely used in scientific discourse with over 3,000 citations so far. And like I said, it's phylogenetically derived. But in reality, like you might have heard of Omicron and Delta and those kind of WHO-based lineages. In this scientific nomenclature system, there have been more than 5,000 such lineages that have been named till date. So if you think about it, on an average, we've been naming roughly 2 lineages per day. And this level of granularity really helps in communicating the exact genetic makeup of the sequence that you are referring to, right? So this particular nomenclature, one thing that we realized when we were looking at this particular application is that for naming new lineages, you actually really care about knowing the exact phylogenetic context of different sequences. So you really want to know like which previous sequences are that we've collected are neighboring to, let's say, a new sequence that has been collected. And also you are interested in knowing which new mutations would have arisen. So this is an example of how we named, for example, the Omicron lineage, which is B11529. Essentially what happened is that it got placed within this B11 subtree, essentially, when it was first discovered, those four sequences which were first discovered for Omicron. This is how the placement would work in that tree-like structure. So that's what this is showing. That's long branch leads to those 4 new sequences that got placed. And what you would notice is on this long branch occurred many significant mutations. So this is how people actually flag this, that this seems to be like a new variant that might have emerged. And you would post it as a GitHub issue, typically, where a bunch of volunteers would examine it and then name it as a new variant that gets designated. So this is how that typically happens. But it's very important for an application like this, like a lineage naming system, that you use comprehensive phylogeny. So you don't really subsample the sequences that have been collected so far. You want to represent all of them, because if you subsample it, you might end up corrupting the naming system. And in some cases, it might also necessitate renaming of variants if you subsample. So this is the insight that we developed our toolkit USHER on. Essentially, what we were noticing is that during the pandemic, we saw an exponential growth in the sequencing of the pathogen. So this is just data showing that within a couple of years, we saw like millions of sequences which were collected from worldwide sequencing efforts. And what we also noticed is that the phylogenetic tree inferences methods that existed at that time were just not able to scale with the rate of incoming data. In fact, we on some days would see tens of thousands to even hundreds of thousands of new sequences being collected. But many of these phylogenetic tools would take more than a day to just have like a phylogeny constructed with a small number of new sequences that are being collected. So they were just not being able to keep up. And in fact, this was an issue raised in one of the commentaries that was made in the Nature journal, where people pointed out that if you really want to track the different variants faster, you should actually fix these type of bioinformatic bottlenecks. Another insight that we gained is that this problem of phylogenetic inference is much better if you use what is called an online approach, which basically involves placement of new sequences on a tree, rather than taking all sequences that have been collected till date and inferring a new phylogeny out of it. It's much better that you sort of freeze the phylogeny that was based out of previous sequences, right? So from previous day, and just incorporate new sequences that were recently collected and just grow that tree, right? So that's the difference between online approach, where you grow the tree, rather than a de novo approach in which you take you assume that there's a static data set and you just de novo infer it from scratch every single tree. So obviously the de novo would be much more efficient than something like an online approach, right, which involves phylogenetic placement. Another thing that we realized is that the SARS-CoV-2 sequences which were being generated tend to be really similar, right? So in this case, I'm actually showing for different variants of SARS-CoV-2 how the spike protein looks like. And as you can see, there are very few sites in which any of these variants have differences. So just a handful of them in this cases. In fact, when we place new sequences on the phylogenetic tree, we often observe that the sequences are placed at an average mutation distance of just like less than one from its nearest neighbor. So they tend to be very similar. And what this means is that you can apply approximations when doing this phylogenetic placement step. Especially approximations like maximum parsimony work really well. They are actually computationally really cheap compared to approaches like maximum likelihood, which are based on complicated statistical techniques and require very intensive computations to be inferred. And in fact, what we observed is that parsimony was nearly equally good as the likelihood. So as you improve the parsimony, the likelihood automatically improved, mostly because the branches on this phylogeny were really short. So based on all these insights, we developed USHER. Essentially, it's a phylogenetic placement tool. What we do is we assume that there's a phylogeny that was generated on the previous day, and on a new day, let's say a new sequence was collected with certain mutations with respect to the reference sequence. And in this case, we assume the first sequence that was collected from Wuhan serves as the reference. So these were the mutations that are there with respect to that sequence. You can actually place it onto the phylogenetic tree using the tree that you had previously inferred and the mutations labeled on that tree itself. And all we do is like a brute force search to determine which node results in the smallest additional number of mutations to explain that new sequence. That's basically what this is doing. So it's actually searching every single node on this tree, realizing that it's best placed at this particular branch. And the resulting tree has this new sequence incorporated, but also the additional mutations that are required to explain that sequence, right? So that is all there is. So we parallelize this on HPC platforms, and two of my undergrads, Cheng and Kyle, were involved in this particular effort. But the most important part to note is that the structure that you see on the left is what we call a mutation annotated tree structure. So it's a tree, but on the different nodes of the tree, we've labeled the mutations that we've inferred to have occurred on that, right? And once we place that new sequence, again, the resulting data structure that you get is also a mutation annotated tree. So you're going from one mat to another mat as you incorporate new sequences, and this makes it really easy to maintain and grow over time, right? So this is basically what was the main idea behind the USHER toolkit. And this is what is used, this particular tool is what is used to maintain the largest and the most comprehensive SARS-CoV-2 phylogeny that is updated daily till date. And the tree is also periodically optimized using another tool that we developed called MAT optimized. And the reason why I need to optimize it because we use a greedy approach for placement. So sometimes you accumulate like suboptimalities and you don't want them to grow over time. So you have to periodically keep optimizing it, and we kept doing that. In fact, when we crossed the milestone of million sequences incorporated on a tree, we actually released this press release, which basically describes that we probably were the first to reach a milestone of a phylogenetic tree being inferred with whole genome sequences at that time, right? And today we have about 60 million sequences of SARS-CoV-2, making it the most sequenced species on Earth. And we still are able to maintain this tree. So essentially, the main contribution of USHER was that it was really fast and memory efficient. In fact, if you compare it to some of the state-of-the-art tools, you will see that it's about 3 orders of magnitude better in both runtime and its peak memory usage. And because it was so fast, we could actually perform real-time analysis where we could allow public health labs to upload sequences onto this interface and get those incorporated onto the phylogenetic tree. And then they could do analysis like the one which I showed you before with Omicron sequences, that once you place it, you see exactly the location in the tree structure. And then you can use it to, for example, name lineages or analyze which lineages, for example, might be in current circulation. And that's exactly how the Omicron variant was discovered, B11529, and those sequences were being analyzed by this particular scientist, Tom Peacock. And since then, like I said, that about 5,000 lineages have been named for SARS-CoV-2 with basically the same approach. But more importantly, this type of big data analysis that we enabled with the help of USHER actually led to many downstream applications. So besides naming lineages, people have started monitoring different circulating lineages in geographical locations by just uploading those onto the USHER platform and seeing which lineages does it belong to just from the placement data itself. Or it has been used also for tracking introductions in different geographical locations. For example, if you see a cluster of infections, let's say in Texas, can you infer that could that cluster have originated from some sequences or some infected person in California, because you've seen that sort of similar sequences before, mostly in California. So you can draw such inferences, which is very useful for public health monitoring efforts. People have used it, the USHER's mutation annotated tree, also for predicting or developing AI models which predict the fitness of different variants. So you take the genome sequence and you can predict what's the fitness score of that particular sequence. So is it more transmissible than some other variant that might be in circulation, for example, right? People have used it to predict, for example, what future mutations might occur in any given sequences, or analyze outbreaks and super spreader events. And also another application I'll talk about later, which is on wastewater surveillance. But the key point I would like to make is that the reason why we got so much of efficiency is this particular data structure called the MAT. And it uses one key idea, that of evolutionary compression, that I'll refer back to later in this talk. Essentially, what this structure is that you have a root sequence, right? A root sequence, which is, let's say, the first sequence that was collected in case of SARS-CoV-2. And all other sequences are basically placed at the tips of this phylogeny with mutations labeled on them. And the interesting property about this structure is that any sequence at the tip of the tree can be derived from this structure, right? So from the root sequence, so if you just trace the path to this tip from the root sequence, you see a bunch of mutations on it. If you apply those mutations, let's say from this tip to the root, there are these 4 mutations. If you apply those four mutations on the root sequence, you basically end up deriving the tip sequence. So essentially, arbitrary number of sequences can be stored with the help of just one sequence at the root and a bunch of mutations that are labeled on this tree, right? And many of these mutations tend to be even shared. So as a result, you actually end up getting lots of compression with this type of approach. So I'll refer back to this idea later on when I discuss pangenome applications. But just to give you an idea, if you had 6 million SARS-CoV-2 sequences, if you represent it with the help of a MAT, you just see that it can be represented with a few hundred megabytes, whereas a FASTA would require a few hundred gigabytes. So it's 100 to 3 orders of 2 to 3 orders of magnitude of improvement. And USHER led to also many other publications, both in my lab and in my collaborators' lab, where we applied it for other applications, including discovering recombinants and tracking them with the help of like some of the visual platforms that we've developed, right? So with that said, maybe I'll move on to another application that I'm going to discuss, which is that of metagenomics. But before I do that, I noticed that there's a question. Yeah.
We talk about freeing up some space for improving speed. Have you observed it in your development of USHER? You mentioned the tree structure with the naming but also it would lead to a lot of complications. Would there be any trade-offs somewhere in between how compact the data structure is and how
Yeah. So that's a very good question. So the question is about if there's a trade-off between the amount that you compress and the speed that you get at the end. And in fact, in this case, what's really special about the way we compressed is that the compression actually leads to more efficiency. So it's actually not a trade-off. The more you compress, the more efficient you are essentially getting in analyzing it. Mostly because the way we compressed it is actually very useful in the way we are doing the placement itself, right? So the MAT is basically like a data structure representation. Exactly, that's what serialized and stored in a file format. So when you actually are performing the placement of new sequences, you exactly use that data structure. So had you used some other approach, which the traditional approach is, which is to first load the multiple sequence alignment and the FASTA, it's a lot more inefficient because first of all, it takes time to load those large file formats, but also you have to still infer the mutations onto the tree again, right, from the alignment. So that inference can also slow you down. So in this case, we store exactly what we need while also compressing. So that's why we call this compressive approach, where you can actually compute efficiently on compressed data. So related application to what I just mentioned in pathogen surveillance, we also developed a new tool, which a preprint of which will soon be available, that of WEPP, which is used in the metagenomic application. Specifically, even within metagenomics, we are looking at this wastewater epidemiology as one of the applications where we are seeing this, lots of growth of genomic data in recent years. And the key idea here is that you're using the sewage. Sewage water or the wastewater to detect traces of infectious diseases. So essentially what you're doing is collecting a sample from, this wastewater and sequencing the fragments of different pathogens that might exist there. And from those sequences, you are sort of inferring which different variants might, for example, be under circulation in a particular community, right? And compared to clinical sequencing, which is the application that I mentioned earlier, where you're, sequencing infected individuals in the clinics, wastewater actually offers several advantages. And one advantage, and some of them are listed here, one such advantage is that it's more cost effective. Rather than sequencing every person who's infected in a community, you're just like trying to use the wastewater data or the sample from the wastewater to just sequence it all at once. It's also less biased, which basically means that you don't have the sampling biases that you typically see in the clinics. those people who have access to the healthcare facilities or who show signs of infection or symptoms, they are the ones who typically get sequenced in the clinical sequencing case. But this is relatively much more unbiased. It's also more timely. What that means is we don't have to wait for a person to show up in the clinic after getting infected. You can just you sequence the wastewater and you immediately know if people are carrying fragments of the pathogen, right? So there are many reasons to be using wastewater epidemiology and many reasons why it's very, very effective. And this is one of the reasons why over 50 countries have actually developed active wastewater epidemiology facilities in recent years. That includes many low and middle income countries. And many of these efforts, of course, began during the COVID-19 pandemic and have since continued, right? And one reason why we think this is going to be a very impactful field is because what we are seeing is that the clinical pathogen sequencing is also on a decline very rapidly. So this is just showing you how many sequences have been deposited over time. And what we are seeing is that since the declared end of the pandemic, we are sequencing actually very little, right? And as a result, it's become much harder to discover new variants from clinical sequencing data. We are also noticing much longer delays and also inaccurate and incomplete information in many cases. So the question that we are posing as a lab is, can wastewater epidemiology help in this particular application, which is to discover variants faster and analyze how the virus is still mutating and so on, right? And one issue that we've noticed with the current bioinformatic tools that exist for wastewater epidemiology is that they are mostly focused on just one application, and that's of lineage abundance, right? So what I mean is, like, we had this 5,000 different lineages of variants that I referred to for SARS-CoV-2 earlier. They are trying to predict which of those are in circulation and in what proportion. So this is basically what is showing. This data is San Diego's wastewater lineage abundance data over time. And as you can see that over time, different lineages die out or take over, start dominating the pandemic, right? So that's basically the pattern that we keep seeing. So people normally try to estimate like which one is growing fast, which variant is going to become important, and so on, right? So the way people do this is now, of course, like in the wastewater, you're going to see a whole mix of variants under circulation. So it's a fairly complicated data to analyze, especially because you don't even see whole sequences being preserved. You actually end up seeing only fragments of the genome, which are completely mixed from different variants in circulation. So people have developed tools which are helpful in deconvolving this type of data, where essentially what they are doing is they are discovering which mutations might be observed in a sample that's, let's say, a vector B, and you're trying to estimate a vector X, which best explains what you observe in the sample through this other information A, which is essentially a matrix of mutational barcodes of different lineages. So essentially, this is USHER derived. What basically they do is that the 5,000 lineages that you have, they have their own signatures of mutations. So that is what is used to construct this matrix A. And if you solve for this deconvolution problem, then you essentially can infer X, which is basically the abundance of different lineages that would be in circulation, right? So this has been a very impactful application during the pandemic. And in fact, one of the leading works in this space was developed at UC San Diego by Rob Knight's and Christian Anderson's group, a tool which is called Freya. But there are some limitations of this type of analysis. One such limitation that we often observe with WB that has a significant gap with clinical sequencing is that of the resolution, right? So what I mean is that they are focused on this lineage level information, which variant might be in circulation, like B117 and so on. But within this lineage, you can observe a lot of genetic diversity that might be important. So here I'm showing you one such example where there's a lineage BA1, and you see two sequences which are collected from the same region clinically, and this is shown on the USHER tree, and you see that there are about 50 mutations that separate them, which is actually quite a lot when you're talking about a pathogen like SARS-CoV-2. So they are not able to discover that level of granularity of different lineages that might be in circulation. So it's low resolution relatively at lineage level. Similarly, you observe that when the lineages are very deep, you are actually having just one signature for the entire lineage. And of course, that results in some inaccuracy. that you observe. And so in many cases, you see that the expected abundance deviates a lot from the estimated abundance for example, simulated studies. Similarly, because you're dependent on the lineages which are already designated for which you have mutational signatures available, it's very hard to discover new variants with the help of wastewater epidemiology. So this means that with now more delays in clinical sequencing efforts, wastewater is also suffering a lot because those delays directly affect the downstream analysis with the wastewater. So to address these limitations, we developed a tool called WEPP recently. It stands for wastewater epidemiology using phylogenetic placements. And the key idea that we use is that in the USHER's mat, which we are already updating daily, so it incorporates all clinical sequences that have been collected till date globally, and they have this lineage labels within them as well. You can actually use phylogenetic placements of the sequencing reads that you collect from the wastewater and determine not just the lineage proportions, which people have been doing already, but also try to infer which haplotype might be present in the wastewater sample from these reads. So essentially, in this example, we are showing that node H1, H2, and H3, each of these nodes correspond to one unique sequence in the mat. They are the ones which probably seem to be present according to WEPP in this particular sample, and their haplotype proportions would be as follows. And because you now have haplotype proportions, you can also derive lineage proportions because you already know H1 and H2 belong to L1. So their combined proportion becomes the proportion of L1, and L2 is basically having just H3 as its corresponding haplotype. So you do haplotype resolution, but also clinical sequencing may not have all the mutations that might have occurred, and you may, because of the delays in sequencing efforts or also sampling issues, you want to be able to discover new new variants with the help of wastewater data. So what WEPP does it also reports what are called unaccounted mutations in WEPP. Essentially, the idea is that these are the mutations that you observe in the wastewater sample, and you cannot explain them just by the haplotypes that you've selected from the mutation annotated tree, right? So these could be indicative of new variants that might have emerged. And besides that, it also reports read to haplotype mapping for individual reads, right? So some reads can also be mapped to multiple haplotypes. And in fact, that's the case for most reads. Essentially, what we are trying to do, if I explain in just one picture, what we do with previous wastewater analysis is like a picture on the left, very blurry analysis where you don't get fine resolution. And with WEPP, what you see is like much finer resolution and discovery of features which are much more detailed and can enable lots of downstream applications as a result, right? Which currently wastewater epidemiology tools lack. So I won't have time to go into details of this algorithm, but I'll just give you a very brief overview. This tool has been developed by 3 of my students, Pranav, Manu, and Pratik. Pratik is at UC Santa Cruz, advised by my collaborator, Corbett Titek. But essentially what this method does is that it takes as input this USHER's mat, right, which has already all the sequences clinically collected, incorporated, and the sequencing reads from the wastewater sample. And then it tries to place, just like USHER places sequences on the tree, it tries to place the sequencing reads on the tree. So this problem is slightly different because the reads are short segments of the genome and not the whole of it, which is why we can apply more optimizations when doing the placement as well. But based on these placement of reads, you can determine which nodes are most likely to be present. So generally, you would give more weights to the nodes which are more number of reads placed, for example. It's a little bit more involved than that, but that's the key idea. And once you do that, you can then select those nodes and apply some similar deconvolution strategy, which I described earlier with Freya, to determine what are the abundances of those haplotypes, for example. And you can repeat this process to refine it. And at the end, you would get all of this data, haplotype proportions, lineage proportions, unaccounted mutations based on just the residue of the deconvolution algorithm formulation. But also you can assign reads to different haplotypes that you've selected. So we have some preliminary results on how this method works. And what we find is that because we are now using comprehensive data as a prior to select the haplotypes, which might be present in the wastewater data, we actually end up seeing that the accuracy of lineage abundance estimates improves dramatically. And as you can see that the RMSE compared to both FRIA and another technique, related technique is much better. In this case, lower is better. It is also able to perform a very important application that so far has only been restricted to clinical sequencing data, and that's to identify intralineage clusters that might exist in a particular region. So here I'm showing an example that we simulated. So these are two clusters that you see in this particular lineage, e.g. 5.1.4. A red cluster and a green cluster. Red one has 4 haplotypes within it. So we simulated, and what we find is that the haplotypes which WEPP picked, those are the ones which are shown in golden stars, they actually match exactly what we had present in the wastewater simulation. So is the case with these two lineages. We find either the exact haplotype or a haplotype that's very close to it within like 1 mutation distance on an average. And so this type of analysis was previously not possible. You would just report that EG 5.1.4 is just 21%. But now with WEPP, you can also discover that there are 2 distinct clusters of this particular variant that are in circulation in this particular region, right? Similarly, you can do analysis of this cluster introductions. Like I said, the transmission networks can be analyzed with the help of WEPP-like techniques, where essentially, because you pick, let's say you collected a wastewater sample from Delaware, and what you found is that there were haplotypes that were selected, these 2, which had 4 and 1% proportions. And when you see this type of placement on the mat, what you observe is that most of the sequences around these 2 haplotypes come from California, right? So when a public health official would see this, they would basically generally infer that it's very likely that this particular cluster in Delaware might have been originated from a place like California, where we've already seen this variant under circulation, right? So this type of analysis can be done. We also notice in real world analysis that WEPP's inferences correlate better with clinical sequencing data. In this case, I've shown the KL divergence of what we see with clinical sequencing data and what we see with WEPP, and they correlate better. Lower is better in this case. So WEPP correlates better than some other approaches. So that's about WEPP. Yeah, before I move on, I'll take the question, and there's one question online as well. Yeah, but I'll take yours first.
The example you gave transmitting from California to Delaware, are there ways to correct for biases? So of course, one way is to normalize for the number of samples taken from places. But are there other biases that could be involved in detecting these that are kind of working on?
So maybe I would need to understand the second part of your question a little better. The biases in what?
So we collected some wastewater samples from wastes, and we detected also something from California has moved to the. Thank you. So yeah, the second part of my question was that when we have some mutations, we observed that they might have moved from California to Delaware. It might just be that we had a lot more samples in California. But one way probably to work around it is to normalize based on number of samples. Yeah. But are there other biases that you have observed in your experiments and computational ways to work around that? Yeah.
So those are both great questions. So maybe I'll answer the second part of the question first, the biases question, where this is an ongoing problem where certain regions sequence a lot more than some other regions, right? So this part is already well known, where not every region of the world is sequencing at similar rates, and certain parts of the world are severely undersampled. So this is definitely a limitation of what we are doing, which is it is more restricted to like if you want to definitely analyze these transmission clusters exactly, you would want that every region samples somewhat uniformly, which is not going to happen. So this is not going to be a perfect approach yet. For example, in the United States, where sequencing efforts have generally been reasonably good, you can apply this to, for example, infer in like between states what sort of transmission patterns occur. And this is the application that we've seen mostly in clinical sequencing data as well. So there's a tool which we developed called Cluster Tracker, which state public health departments are using for this. And of course, you have to take into account the fact that even within states, there's some variation that takes place, but there's no easy way around it. The second part of your question was, how does this differ from approaches, metagenomic approaches that might have been developed for, let's say, genome graphs, right? Is that, yeah. So genome graphs can also be used for this type of applications where you're essentially in a graph structure incorporating every sequence that is present, and then you sort of align reads to it, and then determine which paths are, for example, most common and which haplotypes might be present. You could have done that, except for the scale that we are talking about with SARS-CoV-2. I don't think that people have been able to generate a genome graph as well. So there's no pangenome that exists for which incorporates all sequences at once. So that's been that's one issue. The other thing that we also notice is this ability to place with mutation annotated tree is really efficient. And the reason for that is that you can actually even perform seeding if you want with the help of placement, and that's something that we are going to be developing. But the idea is that you're searching for mutations rather than having full alignments in place, right? So you first align to a reference, you observe which single nucleotide changes are present, and based on that, you actually search in the mat, and that's very sparse, right? Then looking at the whole sequence. So in some ways, I think there is some efficiency advantages that you have. You could have probably done this with genome graphs. We are not aware of any technique which has, or any work which has generated a pangenome which is this large, incorporating all sequences. So the advantage of MAT is we're already maintaining it. And I'll mention later, MAT is a form of pangenome. If you think about it, well, I'll cover that in a later part of the talk. So things that you can do with genome graphs, you can actually do with MATs as well. And in my opinion, I think there are some efficiency advantages that you gain with the evolutionary compression that you do. So I do notice that there's an online question. I'll take that.
So hello. In the first part of WEPP, you mentioned that you also focus on the novel lineages, right? So I was wondering, like, how do you evaluate the accuracy of your method in that sense? Because in the read alignment part, there might be some alignments that do not actually occur in the nature, right? There might be some chimeric alignments that should be like filtered out in that sense. So is there a mechanism that you can like let's say, suggest that you do in your evaluation mechanism in the, let's say, testing the accuracy of your method?
So I assume you're referring to WEPP and not USHER, right? For the
yes,
wastewater.
Yes.
Yeah. So the way we have done our analysis in this case has been mostly related to, so we've used a combination of simulation data and real world data for this type of analysis. A lot more details would be present in the manuscript or the preprint that we are going to soon release. But the way we do it is we first simulate a new variant, which is not present in the mat, and then discover whether the mutations that this new variant carries are flagged as unaccounted mutations in our analysis. And in most cases, we see that those exact mutations do get flagged. In real world analysis, also we perform this. So what we, for example, did is like we tried to find in wastewater samples if Omicron had occurred before like a clinical sequence had detected it, right? And in some cases, we do discover signatures of Omicron very clearly, and those mutations being flagged as well. It's just that in 2 weeks of delay that we saw that clinical sequences also started showing up. And so this gives us more confidence that, this approach is able to, foresee certain variants that you've not found online in advance. Does that answer your question?
Yes.
Okay.
Yes. Thank you so much.
Great. All right. So with this, maybe I'll move on to another application that we've been very excited about recently, which is that of pangenomics. And maybe I'll now speed up in this part of the talk, so since we are running out of time. But the key idea of pangenomics, like I mentioned even earlier, is that it's basically an emerging field which deals with a collection of genomes that would have originated from a single species, right? And of course, this has many applications to study, for example, how a genomic variation can lead to diseases in humans or certain desirable traits in plants and animals, or some type of microbial resistance or drug resistance in different pathogens. But what we see, and like the question had asked, that you could do this type of analysis with the pangenome approach that we've already developed with, for example, graph genomes and so on. But what we typically tend to see is most pangenome analysis till date has only focused on, let's say, tens to hundreds of genomes at a time, and not to the scale that we were talking about with SARS-CoV-2. So in my lab, what we've been really excited about is like, how do we scale it, right? As we keep seeing more and more data, how do we scale this type of analysis to millions of genomes? Yeah. Yeah, I'll talk about it. Yeah. So I'm going to talk about this graph-based representations, right? So that was the question that was posed. So if you look at it, the most popular representation for storing pangenomes, and you have to deal with that when you're referring to scale, because data representation improves or informs both your computational efficiency, but also the type of analysis that you can do with it. And I think there are limitations in both, which I'm going to cover in this slide. So the most popular representation for pangenomes is this graph-based format. Essentially, the idea is that if you have a genome collection, like the one which I've shown on the right, with these 5 sequences, all you do is that when you have segments which are shared between all 5 sequences, they would refer to a single node, and that node corresponds to a sequence as well. And then you will see some subsegments which actually differ between different sequences. So you will have to create divergent edges and corresponding nodes to it, which represent individual or the different genomic segments that you observed, and so on and so forth. As a result, every sequence in this collection will have a unique path in the graph, right? And from that path, you can derive the whole sequence as well. So this is excellent for certain type of applications, in particular for doing sort of read alignment and variant calling like applications. This has been shown to greatly reduce the reference bias that you see when you're using a single sequence as the reference. But in my opinion, there are some limitations to it, like the question had posed. Few of those limitations are, number one, this data structure does not have any evolutionary information contained in it. What I mean is that certain sequences, like I showed, that they tend to be evolutionary related. So you, for example, can describe it in the form of a tree, like how different sequences are related. Here, you don't have that explicitly stored, like which sequences are more similar to each other than others. So each path is unique, but you don't really know which paths are like how they are related in a tree-like structure, right? So that's number one. Secondly, although they are storing variation, they are not really storing mutations, and there's a subtle difference between them. Variation is essentially an outcome of mutations. Mutations are the event that lead to certain genomic changes, and those changes are essentially what we call as the variation, right? Those are the differences in the genome sequences. But mutations is basically the events that led to it. And sometimes you do care about in evolutionary studies the exact ordering of mutations, how they occurred, when they occurred, and so on. And that information is not stored here. Similarly, you can see that they are pretty hard to visualize as well. Sometimes the biological interpretability is very challenging when you have very complicated graphs. And I'm just showing you with like dozens of sequences in this case. But when you have millions of sequences, much harder to visualize it effectively. And since we come from a computer architecture background, some of you have also worked on this problem. You might have noticed that it's actually very hard to accelerate problems related to graph-based structures. For example, doing alignment of a sequence with a graph is fairly challenging, mostly because it introduces irregular memory access patterns, and the parallelism is also pretty irregular, right? But besides, it's also storage-wise inefficient, and I'll show data to prove that they actually don't scale very well with more sequences. So this has been, I think, well known by now in literature that we probably do need more scalable pangenomic representation. So there have been some approaches in developing some alternative pangenomic representations, and these are broadly divided into 2 categories. The first category focuses mostly on compression. So you are trying to represent lots of information, right? So you're still trying to store these large collections of raw genomic sequences and compress it very efficiently. So here the examples are AGC and Minify. You can think of them as more advanced versions of gzip compression, where they are using some biological insights to compress more effectively. But you would notice that in this data structures, you don't store variation information, right? So these are not like graphs or MAT, which also store variation. The second, focus area, which you would see in the literature, is that on representing more information, because as we discussed that in graph-based, some graph-based structures, some information is not present. Examples of that are pangraph, where you also store like the evolutionary relationships. In addition to the graph, you store the evolutionary relationships that the different sequences share. And in tree sequences, you store, for example, more complex mutations, for example, like recombination. That's what this is focused on. So it's a ancestral recombination graph generalization that these approaches store. So if you think of the landscape of the pangenome representations, you will see that there's some sort of a trade-off involved in the memory efficiency and the representative power, right? So those which are highly memory efficient, so take less bytes to store, like Minify and AGC, they represent less information. They don't even store the variation. And the variation preserving graphs, which also store pangraph and the phylogenetic structures and so on, like pangraph and tree sequences, they have high representative power, but in terms of memory efficiency, pretty bad. So what my lab has been trying to work on is to improve this trade-off, right? In fact, what I will show you today improves both the representative power and the memory efficiency. So its memory efficiency is in fact comparable when talking about pangenome data. You will see that its memory efficiency is comparable to the most advanced compression techniques, and it is actually slightly more representative than other formats that have been so far conceived. And the idea that we are going to use is very similar to this evolutionary compression idea that we presented with the MAT, where you store a root sequence and then a bunch of mutations on the phylogeny, and with the help of this, you can essentially derive any sequence on the phylogeny, any sequence corresponding to a node in the phylogeny, by applying the mutations from the path to that node from the root. But there's some limitations in what I had shown earlier with USHER MAT. Specifically, what we had done is that we had a reference that was used at the root sequence, and what we did is every time we placed a new sequence, we compared to that reference, found this single nucleotide mutations, and those were the only ones which are represented. So this has some reference bias issues which the pangenome structures won't have. But more importantly, it ignored indels, right? So it only had substitutions with respect to the reference, but not indels, which was stored in case of the MAT. And these are sometimes very important in doing various analysis. And finally, in phylogenetics, tree is most often used when talking about phylogenies of different pathogens, because we just assume a tree-like topology in the evolutionary pattern, but that's not always the case. In fact, you see complex mutations which involve sometimes two different variants hybridizing to form a new variant. In this case, it's not like a vertical mode of evolution, but it's sort of involves some horizontal transfers between variants as well. So recombination and horizontal gene transfers are two complex types of mutations which violate this vertical mode of evolution that tree-like structures represent, right? So they require a network rather than a tree. So that would be a more accurate representation in those cases. So that's what we are trying to address, that we want to generalize this MAT, or the evolutionary compression idea introduced by MAT, to pangenomics, which gets rid of the reference biases and can store complex mutations as well. So again, in the interest of time, I'm actually not going to discuss this in much detail, but what I would say is, just like MAT, it's both a data structure and file format. And we've released a preprint about this very recently, and the manuscript is currently under review. But essentially what it's doing is it's taking the MAT and adding mutations which include indels. And that is pretty easy. What we do is we represent gaps as special characters in our data structure. So it becomes very easy, and we use the MSA's coordinate system instead of reference sequences coordinate system, right? So that problem gets solved pretty easily. But because these sequences also tend to have structural changes in them, like rearrangements or duplications and so on, you would ideally want to represent those type of mutations as well within the data structure. So for that, what we do is we first find in the entire genome like subsegments which share homology, and they could also mutate. And the mutations could be, for example, insertion of a new block sequence, or deletion of it, or inversion of it, and so on and so forth. So that way we tackle the structural variation part. And finally, like I mentioned, that there are some complex mutations like recombination and horizontal gene transfer, which result in networks rather than trees. And that gets solved by introducing these network edges. So you have a you think of PanMANs as being collection of these tree-like structures in which some nodes of different trees can combine to form the root sequence of another tree, right? So it's mostly a vertical mode of evolution, but it gets violated every once in a while. That's what we assume with PanMANs. But that also can get represented with this new structure, right? So essentially what we do with this format is that we solve the limitations of many of the previous formats that have been conceived. Particularly, it's not just lossless, unlike USHER. So it's lossless, unlike USHER, and it also represents the whole alignment information, which you can derive from this data structure itself. It stores the phylogeny as well as all types of mutations that you observe, right? So it's, in fact, we think that it's the first format which has unified the multiple genome alignments, the phylogeny, and the mutations all in a single format. which makes it information rich, but as I'll show, it's also very compact and scalable, right? So we did this comparison with various microbial species, and we, as you can see in our data sets, they range from a few hundred to a few thousands in different cases. And what you observe is that the PanMAN data structures offers few-fold to a few orders of magnitude improvement in file sizes. And as you can see, the data sets are pretty small, mostly because we are limited by how large of alignments can we construct, as well as how large of phylogenies we can construct in some cases too. But what's more exciting to us is that it also scales with the number of sequences. So what I mean is we've tried on smaller number of relatively small data sets of, let's say, a few thousands of genomes. But what we see is that as we increase the number of sequences in our data set, we actually see that the improvements of PanMAN over other approaches also widens, right? So what this means is when we scale to, let's say, millions of sequences, we will see that this gap would probably be much larger. In fact, we have some analysis that we've done with SARS-CoV-2 data. So in SARS-CoV-2, the good thing is that they don't have they don't have recorded structural changes, right? So you can just have one single MSA as the whole genome alignment representation. That works pretty well. So we developed a tool called TWILIGHT in our lab, which performs an MSA of this whole genome. And then we use that to generate a PanMAN based out of it with 8 million SARS-CoV-2 sequences. And what we found is it only takes 366 megabytes of information, which would otherwise in raw sequence form would be several hundred gigabytes. And if you take the alignment, it would be terabytes of data. So it's actually, compared to the alignment, 3,000-fold more compressed, which is quite remarkable, I think. So like I said, this improvements typically widen when we have more number of sequences. And if you compare even with some of the compression formats that have been introduced recently, that includes BAM, CRAM, AGC, Minify, all of it, PanMAN is actually performing much better. It's actually competent with the most advanced compression techniques. So here I have highlighted Minify and PanMAN as the two columns. In most cases, what we see is that they are like PanMAN is like slightly better, except in few viral on few viral genomes. But even in those cases, when the data sets get larger, it actually performs better and is also more scalable. Now, the nice thing about this is with PanMANs, you can do various type of things because it's such a information-rich format. You can extract out the whole multiple sequence alignment if you like. You can extract out even the graphs that people use. So you because it stores both mutations and variation, you can actually convert PanMAN into a graph. You can extract out variation with respect to any sequence. That's the cool feature of it, not just the root. You can take any sequence and you can trace its part to other sequences. And from there, you can actually derive what variation it has with respect to any sequence in the mat. And you can annotate information on it and provide various type of summaries and so on, right? So lots of cool things that you can do. So far, we've only developed this mostly for microbial genomes, which are relatively simple and have clonal mechanism to evolve. But we are now extending it to more complex genomes like eukaryotic genomes, which includes humans, and also performing GPU acceleration to speed up the construction step. Yeah, so that's basically been the focus of some of the future work that we are going to do in the lab. So maybe I'll quickly switch gears to one final application that's of comparative genomics. I'll make this quick. Essentially, in this particular field, what we are doing is we are sequencing tons of species at this point. And one key application in this area is once you have genome sequences of different species, can we infer how they are evolutionarily related, right? So that's the problem of species tree inference. Given a set of genome sequences of different species, determine how they are related in this tree-like structure, for example, right? So this, in fact, forms the basis of many evolutionary studies downstream. But if you look at how quickly the data has been growing, you can clearly see that it's pretty exponential over time. We now have hundreds to thousands, in fact, of vertebrates being sequenced. And in the next decade or so, it's predicted that we might sequence probably all vertebrates on Earth. And like I mentioned, that these species trees form the basis of many studies, evolutionary studies, whether you're doing reverse genomics, trying to discover which genotype leads to what phenotype, or discover losses of genes and so on. All of these are essentially based out of species trees. So they are very fundamental to downstream tasks. But surprisingly, if you notice that species trees are not fully resolved yet, right? So you would assume that by now we would have figured out how different species are related to each other. But if you look at just the bird phylogeny, for example, in the last decade, you will see that there have been 4 very influential studies that have been published, and they all differ slightly in their order level relationships. So that's what you can see here. And in some cases, they are also by overlapping authors. But what we are seeing is that as we are collecting more and more data, as we are densely sampling these trees and developing new methods, a lot of these debates are also being settled. So one thing that we did recently in the lab is to develop tools which can automate this process. Currently, it's actually very painful to infer the species trees from raw genome data. So what we are trying to ask is, can we automate it in a way that you require no annotations of genes prior to inferring the species trees, don't have to do complicated parameter tuning, which is typically done by domain experts, and which is also accurate and scalable, right? So can we have a computational tool that can go directly from sequences which are being collected to the species trees as the final output? And that can truly democratize phylogenetics, because currently we really have to rely on experts to get very accurate phylogenies. And so that's a tool that we developed recently, led by my student Anshu and collaborators Yatish Mirarab. It's called RORIES. And without going into too much detail, essentially what we've done is remove some of the complex parts which involve annotation and lots of parameter tuning, could require multiple whole genome alignments. We removed those parts from the conventional pipeline and replaced it by a simpler method like random sampling of genes. Surprisingly, this does work when inferring the final species trees. And instead of relying on orthology-based inference, we rely on homology-based. And the key idea here is that when you see copies, the duplicates of the same gene, you don't have to determine exact one-to-one correspondence between them, which one are the exact matches, but you can actually have like a homology inferred, which basically means that it could be through duplication or through speciation. It doesn't matter how they share similarity. So there are some summary techniques which have been recently developed, which we take advantage of to automate this whole process. And with RORIES, we've actually analyzed many data sets so far, including mammals, birds, flies, fishes, and fungi. And on all of this, we've analyzed pretty large data sets and found that the inferences that we get with RORIES are actually pretty comparable in accuracy with domain specialized techniques, right? And in fact, this work was published recently in PNAS and was also featured on the cover of this journal. So since we are running out of time, I'll just quickly advertise that we've also been working in my lab on fundamental methods for sequence analysis, which includes alignment and phylogenetics. I'll just quickly go on it. So with Talco, what we've done is we've sped up pairwise sequence alignment tasks, especially for long read alignment tasks. And the key idea here was that we don't perform a heuristic, but which lose accuracy, but perform a tiling which is fundamentally the same as the full banded techniques, right? So we developed a full accelerator based on this, and in fact, it was one of the best paper nominees at HPCA last year. And we use that to now apply tiling on multiple sequence alignment problem as well. And with that, we are this tool is called TWILIGHT, which we recently released, and it's going to appear at ISMB next month. So the same tiling technique got used, and we parallelized this multiple sequence alignment problem on GPUs. And in fact, this was the one which we used for developing that MSA of 8 million SARS-CoV-2 genomes. And it has support for both AMD and NVIDIA GPUs. And lastly, I'll just mention very quickly, if I may, that we are also using high-level synthesis now for speeding up dynamic programming tasks, which are used very widely in bioinformatics. And for that, we've developed a framework called DPHLS, which allows people to modify the dynamic programming equations onto the front end and get very optimized implementations as the output. And we are also working on some phylogenetic inference techniques, which are distance-based on GPUs, with which we've been able to now scale to millions of sequences, which was otherwise not possible. So with that, I should thank all of you for attending this talk, and I'm happy to take any questions. Yeah.
I think we have time for questions.
Okay. So I assume your first work, USHER, it still is less accurate than a maximum likelihood method like IQ3, right?
Yeah.
So until what extent do you observe how well it scales in terms of accuracy, your method?
Yeah. So the question was like, we are using approximations in USHER, but particularly we're using maximum parsimony, right? So, and plus greedy approximation to that. So how accurate is it? And compared to maximum likelihood approaches, right? So what we have observed is when the phylogeny is very dense, and so you have short branches, right? So this is a well-known thing that when you have very short branches, maximum parsimony tend to work really well. So really what we've conceived USHER for is this type of scenarios where you're not trying to estimate species trees, for example. So what we do with RODIs, where we're estimating species trees, you would not do with maximum parsimony or USHER-like approach, right? What it's really used for is when you have dense phylogenies where sequences which are very similar are essentially being used for inferring the final phylogeny, and the branches are pretty short in that phylogeny. So it's used more for the surveillance, genomic surveillance like approaches. So the question is, how far can we push this? Like, because in certain pathogens too, we see that there could be long branches, right? So far, what we've observed is, so USHER, I didn't mention it, but USHER has now been applied to like close to a dozen pathogens. That includes monkeypox and influenza, RSV, and so on. And it's supported on the online interface. So for such applications, we are seeing that USHER performs pretty reasonably well. It won't be a perfect phylogeny, but for most applications that we do with genomic surveillance, it seems to be okay with that. But you would not, for example, apply it beyond that, which is, for example, analyzing species trees and so on. And even in pathogens which are, for example, evolving too fast, right? That's where you need to be cautious. Yeah.
I see. Also, you compared the MET-based file format to FASTA, and I was wondering why, because you could also compare it to .nwk, I guess, and you're comparing it to FASTA. What's the reasoning behind it? Basically?
Oh, so the reasoning is because the MAT stores at least the same information as what a FASTA does. It stores also the information that a NUX stores, right? The tree structure, right? So it has information which is combined of both of these, right?
Mm-hmm.
In fact, it's storing even the alignment, which is not the raw sequence FASTA. It would be even bigger than the raw sequence FASTA. Right? So it's storing all of this information. So it was just to give you a ballpark estimate that it's doing a compression. And we do a compression of that FASTA too with gzip, and its PanMAN is even more compressible than that on the datasets we observed. So it was just to give you an idea like how good the compression is.
Okay, thank you.
Yep.
Thank you very much for the talk. I have a question regarding the compressed representation of these pangenomes with trees. So to my intuition, graph represent alignment or sequence alignment in a very intuitive way. But these trees seem like when I want to do similar alignment, I have to pay significant overhead, no?
When you want to do alignment to...
yeah, the whole pangenome, basically.
Actually, that's not necessarily the case.
Okay.
So the way you would do it, and we are still developing tools for doing this type of read mapping, which involves indels in it. But for example, if you look at what WEPP does, it's actually performing. So actually, I wouldn't say that WEPP is performing necessarily the alignment because it first does it with the reference. But what you could do with, for example, PanMAT is you can develop a seed table even for the whole pangenome representation in the PanMAT. So that's something that we are still working on currently, where you would, for example, do a read mapping onto the PanMAT itself. So the way you would do it in a graph genome structure, for example, is you would do seeding followed by aligning on the path. Here, what you would do is you would seed to first determine which node does it most closely resemble, right? From just the seed heads, and the seeds would map to the nodes of the PanMAT. Okay. Once you do that, once you identify it, then it's just a pairwise alignment of sequences and not it's sequence to sequence alignment and not to sequence graph alignment. So it actually simplifies it.
Yeah, but okay, I understand, but doesn't that that very much relies on the quality of the seeding, right?
Correct.
Yeah.
But the same is true for genome graphs.
Yeah, sure, in some sense, yeah. But I just feel okay, but probably there's no numbers like.
So what you could achieve, this basically what I'm trying to say with pangenome mats, what you're effectively doing is you're avoiding sequence graph alignment completely, right? Because you're only going to do sequence to sequence. You just have to determine which sequence to align to in that. The analogy to it is which path to align to in the genome graph. And the way you do it in case of genome graphs is that you first map it to the nodes, right? So the seeds are mapping to those nodes. Here also it's mapping to the nodes of the tree. So but those nodes are just just single sequences. They are not paths in any way, right? Okay. So that's the key idea.
Okay, but you do not have any results yet regarding that, right?
Yeah. So that tool is still under development. Yeah. Okay. But I can give you like preliminary idea that what we are finding is it's actually more efficient to do it this way than to do graph alignment.
Okay, and there's no significant loss in accuracy probably then. Okay. Yeah. Thank you very much.
I have one maybe, while maybe others are thinking about questions. So for the RODIs, so you talked about this species level tree inference, right? I'm curious about the challenges that you may expect when you go into strain level, basically inference, especially when the antibiotic resistant type of analysis is needed, right? So do you expect or do you observe, or what sort of challenges that are you expecting in that type of information?
So in some ways, USHER is like strain level analysis tool, because the SARS-CoV-2 lineages have some analogy with the strain data that you talk about in bacteria, for example, right? So in fact, now USHER is also being used for representing many bacterial genomes. You could apply, for example, RODIs even at subspecies level. It doesn't work as well because when you randomly sample it, the different segments, you may not encounter informative sites within it, right? So that's why you would actually tend to use as much of the genome as you can to determine like informative sites within them. That's what USHER basically does, right? So USHER-like approaches can be used. We are working on incorporating some more accurate analysis within USHER, so likelihood-like approaches. In fact, I was involved in one such tool development called Maple, which uses a technique called parsimonious likelihood. So it mixes up parsimony and maximum likelihood to form an approximation. And that also scales much better than maximum likelihood approaches, but would be more accurate than what USHER is. So when you have divergent strains, very divergent strains within a bacteria, then maybe you would want to use that approach rather than USHER. Nice. Yeah.
I guess one benefit of USHER is you can also do updates to the data structure.
So is the case with Maple. Maple also has placement. Yeah.
Is it the case also with PanMAN? I guess.
So we are developing placements now.
I see. Okay.
Yeah. So right now it's de novo generated, but yeah, we plan to work on placement with PanMAN.
So I really enjoyed learning about the HLS effort that you've done, and I'm wondering what are your opinions about what we can do in hardware community to facilitate the adoption of these types of accelerations in genomic community? Of course, ease of programmability is one thing, but are there other aspects or other parts of the pipeline that you think are in the next level in criticality after the dynamic programming? And other types of efforts other than automation?
So one of the motivations why we developed DPHLS was for that purpose, which is for improving adoption in the community. So I think one thing which the bioinformatics community typically struggles with is the complexity of developing an accelerator or using it, right? They're familiar with C++, but they may not be familiar with Verilog and how to interface bit streams and things like that. It's just too complex of a task. So what DPHLS is doing is it's giving more like a software, like a experience for hardware development. So essentially what you do is you just write your kernels in C++, and there are backend optimizations, which the user doesn't get to see, but they map it to a systolic array onto the FPGAs, right? So that way, this could be one mechanism which we have worked in this direction, that hopefully it will enable more adoption in the community. FPGAs are being used in bioinformatics. Erico, for example, Illumina's subsidiary, has developed FPGA accelerators, which many people have been using. But people also want ways in which they can implement new kernels, right? For example, I want to do protein alignment or dynamic time warping, other applications which are not easy to develop, although they will map very efficiently onto FPGAs, but it's hard to do it. So you want to create levels of abstraction for them so that it becomes more accessible. So that's one way to do this, right? Also, we are seeing in cloud that the FPGAs are now much more easily accessible, and lots of bioinformatics community is doing a lot of cloud processing as well. The other direction that we have been also thinking is also to leverage GPUs. So FPGAs may be suitable for certain applications like dynamic programming and so on, but they're not always most efficient for other types of bioinformatic analysis that you do, right? So phylogenetics, for example, I've not seen a lot of work done on FPGAs so far, but I've seen GPU accelerators for it, and it makes sense for likelihood computations. It would be far more efficient. And with bioinformatics community adopting more and more of GPUs, it's probably like easier for them to expand to more applications if you develop GPU accelerated tools. So that's another direction that we are taking. So I don't know if that fully answers your question,
Yes.
but I guess what I was trying to imply is we do want to improve design productivity with hardware, and I think that's something we should really think about as a community.
Right. And another direction to complement that, I think, would be to think if we were to have a hardware that allows us to program different genomic applications, how would that hardware look like? Is FPGA or GPU the right hardware? Or like, given the importance or rising importance of genomics, could we have, let's say, GPU, like genomic processing unit, that allows us to
so
probably would need to name it something else,
yes.
but
so I am kind of bullish about that idea, but maybe for the long term, where you see data volumes, they keep rising, and at a certain inflection point, people will start taking it a lot more seriously, that we need to have like a lot more specialized chip for it. Right now, I think the economics don't fully support it, the scale at which we are at. That we developed something totally custom like a GPU process or genomics processing unit for it. But I do see that maybe in a decade or so, like in the next decade or so, we will see us reaching that point, just because you're not going to keep seeing gains with generalized processes, right? So you will have to introduce more and more specializations. The other thing which I think maybe as a community we could think about is, can we make GPUs more specialized for genomics? So they are becoming more specialized for AI, right? And GPUs are way easier to program than FPGAs. So that's another idea that I'm quite interested in and very supportive of. So in fact, with NVIDIA's pptx instruction, something that we had proposed a very related idea in our CACM paper on domain-specific hardware accelerators, which I wrote with Bill Daly. The whole idea there was that we could get ASIC-like gains in dynamic programming if we introduce a new instruction in it, right? So that could be another approach that we as a community can think about, that if it's too far-fetched to have a totally specialized processor for genomics, then can we specialize our current processors more towards genomics, right? So that would be maybe an easier solution to get to first.
Yeah. Thank you so much.
Great. Maybe we can take the other questions offline. And thanks, Yatish, for coming. And let's thank the speaker again.
Thank you.